I'd like to thank the organizing committee for the Cancer Genomics Consortium for the opportunity to be here and to present to you some of the newest data that we have regarding essentially the use of long-read sequencing. I want to thank Stuart in particular. I'm sorry I couldn't be with you there today. I was really looking forward to actually personal interactions. Given circumstances, this will be all obviously a virtual presentation. So I think we all appreciate that whenever we analyze a genome, there's in fact a very wide spectrum of genetic variation, ranging from single base pair changes all the way up to large chromosomal events that have typically been the purview of cytogenetics. Of course, our ability to detect this type of variation from the small to the large has largely been driven by advances in technology. Cytogenetics and microarrays have been particularly important for larger events and systematically identifying them in genomes as well as pathogenic material, while sequencing technology is focused on smaller events, detecting essentially small-scale variation within genomes. I would argue that there is, in fact, a lot of genetic variation that has been under-ascertained over the last three decades of human genetics research. And that is largely variation of indels all the way up to large-scale structural variants. So operationally, in the distinction between insertion deletion events and structural variation events is events of 50 base pairs and above, referred to as structural variation events. Of course, the challenge for any genome, but particularly the human genome, is essentially its complexity with respect to repeat structure. And so shown here is one version of that complexity, showing you the pattern in blue of the largest and most identical duplications, called segmental duplications that exist in our genome. What you're seeing here with these blue joining lines are segments that are greater than 20 kilobases in size and greater than 95%. And about 4% of our genome is made of this material. If you add the interchromosomal pattern on top of that, there's about 25,000 pairwise alignments that constitute a human genome, where most of these duplications have arisen relatively recently during evolution. In addition to this, there are areas which I often refer to as sequence non grata, areas around centromeres, indicated here in purple, or in these acrocentrics, the rDNA and short arms of these chromosomes, which haven't been sequenced to date since really the last 20 years of the Human Genome Project. So these areas of the genome are particularly important, both from an evolutionary perspective, because they rapidly evolve and lead to the emergence of new genes, but from a structural perspective in terms of rearrangements. So for example, we have shown, and others as well, that about 49% of all normal copy number changes that exist in germline genomes are in fact mapped to this roughly 4% of the genetic code. So the challenges for actually characterizing variation is really capturing really this more complex variation. And when variation is enriched or maps to repetitive regions, it becomes particularly problematic. So what are the issues? Well, the genome is complex. In many cases, we still haven't sequenced really a good representation of some regions of the genome even once. Short read methods really shine the light on relatively unique areas of the genome. So it's about 83 to 85% of our genetic code, which can be systematically ascertained, but it's particularly good for finding smaller variations, single nucleotide changes, insertion and deletions. Moreover, there are still technological limitations in addition to the mappability biases in terms of, for example, AT-rich and GC-rich regions being underrepresented, particularly with platforms such as Illumina. If we don't discover those variants, we obviously miss on genetic causes of disease. If we don't discover the variants and we don't sequence resolve them, it becomes very difficult to impute and to associate with disease. So I'm going to argue today, the premise of my talk is that in fact, long-read sequencing has introduced a new revolution, I would say, a really fundamental one in terms of how we think about genomes, how we understand variation. And although I'm going to be speaking mainly about germline variation, I think many of the things I'm going to discuss today will be very relevant to individuals working on cancer genomes. So just to kind of introduce you to the technologies, there are two primarily ones that are used quite frequently. There's a technology developed by PacBio, or Pacific Biosciences, which involves essentially what is called a zero-mode waveguide, which are these tiny slits in which single DNA molecules actually enter in, so native DNA. A polymerase that's been engineered sits at the bottom of the well. So So this is the zero mode wave guide. And fluorescently labeled DNTPs are directly incorporated. As they are incorporated, both laser excitation and emission is detected, allowing for the direct incorporation of nucleotides to be recorded. There are two different modalities for PacBio. There is the continuous long read approach, where you can generate sequence reads about 50 kilobases or even a little bit larger in size. And then there's the circular consensus modality, which I'll show in another slide, which allows you to go around a molecule multiple times to generate fairly accurate, high fidelity long read sequence. The other technology is Oxford Nanopore technology. And I'm showing you one of their three platforms, which is the GridION. But essentially, they have a completely different chemistry where they, in fact, long DNA molecules are brought by a motor protein. They are unwound to a pore. And the pore protein then, the DNA single strand is going through it. And as it goes through, this is a membrane that has ion potential across it. And as that ion potential changes, they can infer the actual sequence. Both single pass technologies have a relatively high error rate, about 10%. And so I would argue that the two advances over the last year or two that really have, I think, made these technologies much more amenable and useful to geneticists and cytogeneticists is essentially the development of really HiFi PacBio sequencing. So that's high fidelity. And here the idea is pretty straightforward. You put these what they call smart bell adapters, which are essentially hairpins at the ends of the molecules, and such as the polymerase moves around, it goes around single passes multiple times, creating subreads. Based on those subreads, you then generate a consensus of sequence, which is highly accurate. Some estimates are 99.9 to 99.9% accurate 18 kilobase reads. The second really is the development of ultra-long read technology. So in particular for Oxford Nanopore, there seems to be no limit to essentially the molecule that can pass through a pore. Molecules in excess of 2 megabases now have been shown to pass through the pore. It only depends upon your prep. So if you have the time to generate essentially particularly high molecular weight DNA, you can sequence it with ultra-long read technology. And you can generate DNA profiles, and these are sequence read profiles here, where the average mean read length is about 60 to 100 kilobases in size, which generates reads that are over a megabase in size. Both of these advances, I think, have been game changers, and I hope to show you that as we go along today in today's talk. So my objectives today really are four. One is to really show you where we started with this in terms of using it to understand structural variation, really at the sequence base pair level within genomes. I'm then going to move to some more recent work that we've done in collaboration with the Telomere Consortium to sequence and assemble chromosomes from telomere to telomere for the first time, and what we've learned from that. And then I'm going to talk about really quite a hot area these days, which is essentially the building of phased and sequence assembled human genomes, where instead of thinking about a genome as 3 gigabases of sequence, we're now thinking about it in terms of 6 gigabases, and how I believe this is going to revolutionize how we characterize genetic variation going forward. And then I'm going to end with some applications and with respect to both disease and evolution. So we got involved with long read sequencing technology on the order of about 6 or 7 years ago. And when we did this, we decided that we would apply it to the sequencing of a human genome. And we didn't just pick any old human genome. We took a genome that was essentially haploid in nature. So some of you are familiar with complete hydatidiform molds. These are samples in which you can think of it as an enucleated egg being essentially fertilized by a sperm. And in essence, the maternal complement is typically jettisoned and the actual male complement becomes diploidized. But as a result, you create a cell line, or a cell line is created as a result of this error of reproduction, in which there's essentially only one complement or one haplotype present. So why did we want to start with something like this? Well, a lot of my lab's research over the last 2 decades is focused on studying duplicated sequences. And some of these duplications are so highly identical, the only way to tease them apart from alleles is to really separate the haplotypes. And so this provided us a way whenever we would see sequence differences to be absolutely sure that we, in fact, were looking at paralogs or duplicates as opposed to the original, as opposed to alleles. So a postdoc at that time, Mark Chasen, who's now an independent faculty at UCSC, developed an approach based on essentially an old aligner that he had developed called Blazer, which would align long-read sequences, align these sequences, these long-read sequences that we generated. I think in this case, they were about 12 kilobases in size, and actually look for signatures that would be indicative of structural variation. So areas where it looked like there was additional sequence, or the sequence would all stop on one side, or there was evidence of inversion. Once you had identified them, then go back and pull out those long-read sequences, do a local assembly over the region, build a high-quality consensus or a contig of hundreds of kilobases in size, and then map that back to the human reference, defining the breakpoints. So Mark did this for this first hydatidiform mole. And what I'm showing you here is his discovery with respect to the human reference. So everything that you see in black or gray means that there was an insertion in that hydatidiform mole compared to the reference. And I've broken it down in sizes and 100 base pair bins, starting at roughly around 30 base pairs, up to 1,000 base pairs here, and then from 1,000 to 20,000 on the second plot. Everywhere you see that's pink or red, this represents areas that are deleted, so they're not present in the reference, or they're deleted, I should say, in the hydatidiform mole with respect to the reference. So a couple of things you should note. Right away, you can see that there's this kind of exponential decay, but there are these peaks at essentially 300 base pairs and 6 kilobases in size. This is essentially reassuring because this corresponds to ALUs that are mobile elements in our genome, or L1HS elements, which are line elements that are obviously different between individuals because of retrotransposition events. Now, everything that you see in dark black or dark red is significant. What I'm showing by that is that these are sequence variants, structural variants that had never been seen before in over thousands of human genomes that have been studied with Illumina short-read data. Those indicated in gray and pink are ones that were overlapping known events. So when we did this analysis, the surprising thing, even to us, was that 92% of the insertions, 60% of the deletions were essentially novel compared to anything that had been previously reported. So just to add up the bases, that's about 22,000 novel genetic variants corresponding to about 11 megabases. In this same genome, there's about 3.3 million single nucleotide variants corresponding to 3.3 megabases of sequence difference. So there is 6 times or 5 times the amount of variation that was present in these genomes as a result of structural variation had been previously unascertained, including variation in exons, promoters, and regulatory elements. So once Mark had basically characterized the first 2 hydatidiform moles, we then turned our attention to diploid genomes. And we used the actual haploid genomes to essentially model what we would miss or detect. And we found that we would miss about 30% of structural variation if we just went kind of blithely into and generated long-read sequence. So we came up with a very simplistic strategy, which was this, and this was in collaboration with a colleague in Germany, Tobias Marshall, which was to take human genomes and essentially convert them into 2 haploid genomes. And the way we would do this, we would take a diploid human genome and we would sequence it with long reads. But then we would actually phase the reads, partition them based on Strandseq sequencing data, 10x sequencing data, or with HiC data. So we'd use the sequence differences that exist in that genome to in principle phase. So Strandseq or strand sequencing is a technology by which you basically take a cell, you incorporate BRD label, but you destroy one of the 2 strands. And so you actually generate short read sequences that are essentially physically linked from telomere to telomere as a result. So this was actually technology developed by Peter Lansdorp out of Utrecht and Vancouver. So once you had done this, and you had generated the long read sequence, you could essentially partition the reads. You could do the exact same experiment that you did before, mapping the long read sequences back to the human genome, but then partition them into either haplotype A or B based on this data, and then locally assemble. So we did this for 3 parent-child trios. So this was a Yoruban, a Puerto Rican, and a Han Chinese. We sequenced each of those genomes with Illumina. We applied structural variation collars designed to detect from Illumina sequence. We did long read sequencing, and we did phasing as described using Strandseq data. And we also did one other technology, optical mapping, which provided us kind of a third technology to compare. So what I'm showing you are the results from these experiments, comparing the different sequencing platforms. So everything that's below the line is essentially deletion. Everything above the line is, in fact, an insertion. I think you can see right away that the blue lines are well above the red. So this is the increased sensitivity. In fact, if you quantify it, 30,000 PacBio variants were detected, somewhere between 25 to 30,000 per individual, compared to 11,000 detected by Illumina. So about 3 to 1. If you broke it down in terms of deletions and insertions were woefully underrepresented. So typically 90% of insertions would be missed, and about 50% of the deletions were missed by Illumina. The other thing I want you to note, which I think is an important point and will be relevant when I discuss telomere to telomere sequencing, is that you can see that there is a portion of these genomes which is not detected. It's only a couple hundred variants, but it's for very large structural variants, greater than 10 kilobases, which are being detected by optical mapping, but not by the sequencing technology. But overall, when we put it all together, we estimated somewhere between only 25 to 27% of all structural variation is being detected when you sequence a genome with short read sequencing technology. So as a corollary of this work were efforts to take those same hydatidiform moles that we had described began to work on in 2015. And to begin to do, this is work largely of Adam Philippi at NIH and Karen Miga at UCSC, which was to try to build telomere to telomere assemblies of chromosomes. So in 2020, there was the genome sequence was released. And this is showing you, anywhere you see a transition from black to gray means that this is a break or a contig break. But the important point is that this sequencing of the hydatidiform mole, that majority of the gaps were essentially closed. There's only a couple hundred gaps that remain in these genomes, including, in fact, some centromeres being fully sequenced and assembled. Now, while this takes additional work, there are efforts that have been led to actually sequence from telomere to telomere. The first one was just published last week. It's the X chromosome. It's published in Nature. And I'm going to share with you our own work on closing the first autosome from telomere to telomere, including all duplications as well as the centromere. So what is the approach? Well, while this is still work in progress, the general outline of the approach to generate telomere to telomeres, at least from hydatidiforms, is to generate ultra-long sequence. So one set of data, which is the ultra-long ONT data, where you have your mean at about 100 kilobases in terms of the size. Generate from the same source HiFi PacBio data. This is typically between 15 to 18 kilobases, but is highly accurate at 99.9%. Then to take your ultra-long data and to identify sequence differences that in fact exist once and only once in the genome. So we call them SUNCs, for singly unique nucleotide identifying k-mers, and use that to encode essentially the ultra-long reads. Once you have them, even in repetitive regions such as centromeres, it becomes possible to traverse those sequences if the molecules are sequenced and are present at a high enough depth. So this is in principle, and you would use the ultra-long reads as well as the spacing between them to basically be confident of their location. Of course, that will generate a scaffold genome assembly, which is only 87 to 98% accurate, depending upon the region that you're looking at. And then to replace those, once you've created the sequence-based scaffold, with essentially the high accuracy PacBio data. So this is 99.99% accurate as a final product. So this is the sequencing of chromosome 8, at least from this hydatidiform mole, all 146.3 megabases as a contiguous sequence from telomere to telomere. Telomeres are indicated in these dot plots here, about 1 to 2 kilobases of T2A triple G sequences, with degenerate sequences located immediately proximal and distal. There's another complicated region that some of you may be aware. It's this roughly 6 megabase region, which contains the defensin cluster as well as an inversion polymorphism. We were able to sequence it completely, including a 544 kilobase duplication that is duplicated 3 times in this particular sample. It includes a gene family called defensins, which increases in copy number and decreases associated with chronic inflammatory diseases in the human population. Some of you may also be familiar with the fact that this region also corresponds to the largest common inversion polymorphism in the human species. It's about a 4 megabase inversion polymorphism that actually predisposes to rearrangements associated with congenital heart defects in the human species. So in this particular case, the hydatidiform mole contained one of the CHM13 contained one of the higher copy numbers, but we were able to traverse all the way through it as part of our sequencing and validated organization. Probably the apex and the most work in terms of resolving was the centromere. We found there were 11 Oxford nanopore reads that traversed it in its entirety. And these were the unique k-mers which actually allowed us to create a scaffold sequence map of the region. We then did HiFi sequencing and generated a high quality sequence through the centromere. So this was our kind of first layer annotation. The sequence itself of the satellite of the centromere, as you know, is consisting of a centromere, a 171-mer that's repeated in tandem. This, in fact, forms a higher order structure of a higher order array that's repeated multiple times. There are various higher order repeat arrays that were identified in this area, including the ones that have been documented previously by Hunt Willard and colleagues. And as we move to the monomeric ends of the chromosome, you can see that these are areas that are interdigitated between satellite as well as other classes of repeat elements. So how do we know that this is correctly assembled? Well, you go back to old-fashioned pulse fields, and so this is work of Mikhail Liskovich at NIH. And this is showing the pulse field gel southern of essentially the centromere of chromosome 8. And it actually perfectly maps with these 2 restrictions maps of BAMH1 and MFE1. So what did we learn about the organization? So what I'm showing you here, once again, is that schematic of the organization. So 2.08 megabases of alpha satellite in this individual. What I'm indicating here with this dot matrix or this heat map is basically taking this entire segment, because it is repetitive and is tandem, breaking it into 5 kilobases and comparing it with all other 5 kilobases. And so what you can clearly see here is with the warmer colors, it means higher sequence identity. So these in purple and blue are about 70 to 80%. And as you move to higher sequence identity, this moves up to 100%. And what you can see here, I think very clearly, is that you have this interior portion of the alpha satellite corresponding to the center of the higher order. You have divergent sequences on the periphery. And what's interesting is that this sequence, this monomeric sequence, is nearly identical in sequence structure, or is more identical to that than it is to the interior. You see the same thing occurring for this little pocket of high identity satellite sequence just on the border of the monomeric sequence. And you can basically see this pattern of both younger elements going toward older elements, but also a mirror symmetry as well as a layered nature. So we have layers for this monomeric portion, then this portion, which are more identical, and then this interior portion. So in this particular case, mirrored from the younger elements out. And this is very similar to the model that was proposed by George Smith back in the 1970s in terms of how you would expect to see a centromeres organized. So that's essentially one of the successes for the first autosome that has been sequenced and assembled. And I should point out that the work continues, and it looks as though maybe at this point about half of the chromosomes are now complete. At least we have draft telomere to telomere assemblies. And it's likely that within the year we will have the first completed telomere to telomere assembly of the human genome. But of course, all of this has been based on hydatidiform moles, which is a bit of a cheat because it doesn't actually have the complexity of a diploid genome. So how can that be resolved? Well, cueing from what I presented earlier in terms of thinking about how to resolve structural variation, the same basic approach has been applied to thinking about phasing and assembling human genomes. And so the idea is pretty straightforward. Once again, you start with a diploid genome. You do high fidelity, so this is that circular consensus biosequencing, and you actually generate essentially contigs that are highly accurate from this. At the same time that you do this, you generate linking or phasing information, and that can come from different sources. It can come from HiFi data generated from the same sample, Strandseq data generated from the same sample, or even parent-child trio data, which will allow you to essentially phase. So you generate these contigs, use these high-quality contigs to identify single nucleotide variants. Then you project the actual phasing information on it, and you use that to either phase the reads or directly phase the contigs, such that you now partition a genome from a 3 gigabase assembly to one of 6 gigabases. So you either partition the reads before you assemble, or you generate the assembly first in 6 gigabases, and then you use this information to say what is paternal and maternal haplotype. There are about 2 or 3 formulas that are currently being compared right now with using different orthogonal technologies, but essentially at the end of the day are giving you the same product. So we started on a project to go after one human genome that was diploid. We took parent-child information from a 1,000 genome sample from a Puerto Rican. We generated about 30-fold sequence coverage from each of the 3 individuals. From this trio, we generated Strandseq data, which I mentioned before, allows us to essentially phase from telomere to telomere, provides us kind of a light or sparse phasing information across it. We generated a HiFi sequence. We assembled it using, in this case, a tool known as HiCanoe. And then we essentially used the Strandseq data to cluster these sequences into paternal and maternal haplotypes. 95% of the SNPs were phased, and 81% of the CCS reads that were generated were assigned either to haplotype 1 or haplotype 2. So this is a 6 gigabase genome that we generated. The N50 contig lengths are between 25 and 28 for H1 and H2. So that means half the genome that was assembled and phased was actually in contigs of 25 megabases or greater. And I'm showing you the 2 different, how well we actually assembled and phased haplotype 1 and haplotype 2 from this Puerto Rican. Anywhere you see is contiguous blue or contiguous yellow means that these are either the paternal or maternal haplotype correctly phased. Anywhere we see blue and yellow juxtaposing, such as here, is a potential error. So based on our assessment, we estimate that about 99.8% of this genome, at least the euchromatic portions, were correctly phased. There are still portions, obviously the acrocentrics and the centromeres, which are not phased, but essentially we have created a phased 6 gigabase assembly of a human genome. So once you have it, you can now discover variants, all variants, whether it's SNPs, structural variants, or indels, systematically by comparing each haplotype against human reference. So you're no longer using reads and trying to infer from reads mapping to a reference. You're now using essentially genome to genome comparisons to discover structural variation. So we can discover insertion deletion events, several 100,000 of them, or larger or even larger structural variation events, including the ALUs and L1 insertions in these data. We can then project this onto a map. So this is showing you the structural variation projected onto haplotype 1 and haplotype 2. These are the SNPs. So you can clearly see, and what I'm showing you here are counts of SNPs. There's a total of 4.1 million single nucleotide variants, 2.8 million per haplotype. And you can see areas that are cold, like as expected, like the X chromosome, which has less human genetic diversity, and areas that are hot, like HLA region. So this is the major histocompatibility complex region. And in fact, if you zoom in on those regions are represented as 2 contiguous, complete 6 megabase contigs present in these assemblies. Obviously, this is incredibly important for immunotherapy, since understanding the complete HLA complement is important for developing and customizing immunotherapies down there. So what are the applications of this technology? Well, the first, and I think most important, the one I'm most excited about, is using it to understand chromosome structure and function. I think no better example could be shown than the centromere. Having the complete sequence of the centromere allows us to ask new questions. So for example, one of the things you get, and you get solely from Oxford Nanopore technology, is methylation data. So shown here in red are the methylated regions that we identified across the centromere, and in blue are the unmethylated regions. And you can see that most of the centromere is essentially highly methylated. There is, however, a pocket of about 76 kilobases where the methylation is in fact sparse, suggesting it's an under or hypomethylated region of the centromere. So we were wondering if this actually corresponds to the location of the kinetochore. So this is the only area where we would actually have CenPP boxes in which there are in fact no methylation. And in fact, in some results that I got from my postdoc this morning, a CenPE binding is actually localizing precisely to the same 76 kilobase methylation dip. So these hypomethylated regions is something that we've seen on all the chromosomes to date that we've actually sequenced through the centromere. And I think they're allowing us to define precisely where the centromere function and segregation initiates on a chromosome. One thing I'll point out, showing here is the higher order structure, kind of encoded as colored. The area that we've identified is actually a mixture of different higher order structures. Although there's one shown here in red that predominates, it seems to be that this particular area is in fact very diverse with respect to higher order structure. The other thing you can do is that once you've sequenced one centromere, in this case a human, you can go off and study that same centromere in multiple species. So using the HiFi sequencing protocol that we just described, we've recently generated data from chimpanzee, orangutan, and macaque. And we use this to build for the first time a comparative map of the sequence structure and organization from end to end of each of these centromeres. And maybe not surprising to some people, but in fact, the centromere structure is in fact radically different between different primates. And in particular, the centromeres in macaque are much larger than what we've seen in humans. But once you have this type of data, which is complete sequence, you can now actually build phylogenetic trees. And so one of the things that we've learned is that the higher order sequences, the higher order structures that define centromeres actually evolved in the common ancestor of all greater apes. The trajectory leading to both macaques is completely different. So these actually group in separate clades, but they group more closely to the monomeric sequences we saw at the periphery. And the data from the phylogenetic analysis is also confirming that there are in fact multiple layers. So in this particular case, this is chromosome 8. This was the two different monomeric layers that I showed previously, suggesting that they evolved independently and at different time points during evolution. Of course, for the human genetic clinical community, long-read sequencing has started to enter essentially the clinical area. For those of you who are involved in Mendelian genetic programs, there's a large number, about 50% of cases that remain unsolved. There are also cases where one allele is discovered in a recessive trait, but the other allele is not. And so shown here is just a snapshot of a few diseases where either the disease-causing locus was discovered or the disease-causing locus was in fact completely sequence characterized for the first time. And so shown here are a subset of those. One I'll just mention, for example, this one, TAF1. This was a mobile element insertion that occurred in a gene called TAF1. It was in a founder for a population from the Philippines in which X-linked dystonia Parkinsonism had been segregating for many generations. Here's another one. This is actually developed work from Aisha Ura from Japan. There were 60-some families that in fact, for years, for over a decade, had not been able to identify the disease-causing allele. And they found that in 60 of those 63 families, there was a TTT or a pentanucleotide expansion. In fact, many of the events that are being associated now with new diseases and new disorders were not in the human reference genome to begin with. They were not detectable by whole genome sequencing or by whole exome sequencing. So here's some work locally from University of Washington. This is characterization of patients with Baratela-Scott syndrome. This is a southern blot. For those of you who don't know, some pathogenic alleles of Baratela-Scott arise by hyperexpansion of a CCG repeat in the 5' UTR. This is showing you that in this child who has 2 expanded alleles. Light sequencing actually not only identifies the sequence structure of both alleles, but also in the case of the mother, you can see the premutation allele that expanded from B to D to lead to the disease state. Another exciting area of development is essentially using long-read sequencing to better understand human genetic diversity. So there's an initiative that was launched about just a year ago as part of, it's called the Human Pangenome Reference Consortium, or HPRC, whose goal it is to generate telomere to telomere assemblies of 350 human genomes that represent the diversity, or better represent the diversity, of the human species. And that's supposed to be achieved over the next 4 years. This would in principle generate 700 haplotypes if we're successful from end to end. It's a large group of investigators that are taking on this effort. In anticipation of this, we began our own kind of small project to tackle 15 human genomes. We focused just on structural variation, sampling individuals both from different continents, identified about 100,000 structural variants, which we were able to sequence resolve. There's a couple interesting findings from this work. The first is that we could see that as we sequence more samples, the amount of new structural variants that we would detect that were common is on the order of a couple thousand per genome. So we're starting to, we think, begin to saturate. What's interesting is if we ask the question and look specifically at those that are seen in all humans but are not present in the human reference, there are about 2,000 structural variants, largely insertions and deletions, where the human genome is either wrong or represents the minor allele, and about 13% of the structural variants, which are seen in the majority of the human genomes, but not in the reference. So this necessitates really the development of a pan-genome reference, where we'll be able to actually better represent diversity for doing genotyping later on. Excitingly, if you take the data once sequence resolved, you can go back to essentially Illumina whole genome shotgun sequence data. This is data from the Human Genome Diversity Project. And you can genotype about 65% of the variants that we sequence resolve. So we can go back and make new associations, both eQTLs as well as genome-wide association signals. We've also learned that the distribution of different classes of variants is non-randomly distributed. So for example, we know that copy number variance is more localized toward areas flanked by segmental duplications, particularly large ones. But we now know that VNTRs, in particular expanded VNTRs, shown here in green, about 50% of them are mapping in the last 5 megabases of human chromosomes. We think because these are preferential sites of double-strand breakage. Phasing and assembling of both human and non-human genomes is identifying areas that appear to be recurrently toggling back and forth from an inverted to a direct orientation. So there are areas of the genome that appear to have toggled back and forth, where some humans will look more like a chimp with respect to their organization, where the region, because it's been flanked by segmental duplications, is prone to unequal crossing over at a higher frequency. We've been now able to look for the first time at the sequence composition of VNTRs and STRs. In particular, this is showing you one locus and the variability in the length of a tetranucleotide AAAG repeat sequence. But what's important, it allows us to distinguish those alleles which are continuous versus those that are interrupted. And the length of the longest tract of uninterrupted repeats is in many studies has been shown to be predictive of genomic instability. And so based on our initial survey, really at this point of really a small number of haplotypes, we can predict particular VNTRs and STRs that are going to be capable of, likely to be unstable as a result of their purity in the population. So I'll just leave you with this last thought. In addition to really informing on terms of genome structure and disease, the ability to basically sequence and assemble these complex regions of the genome, I believe, is going to provide fundamental insights as how we evolved as a species. So some of you may be familiar with this particular locus. This is one of the most common causes of autism in the human population. It's due to a recurrent microdeletion and microduplication of about 28 genes flanked by segmental duplication shown here in green. And this is constantly occurring such that about 1% of all individuals with autism in the human population that carry autism have this particular deletion or duplication. So what we determined was that, in fact, this variation that we see, this particular organization of these green duplications, is a fairly recent phenomena in human evolution. So shown here is the copy number of that green segment in different human populations compared to all non-human apes. And what's particularly remarkable, you notice that even between Neanderthal and Denisova, this increase in copy number, which are additional copies of this green segment, is found exclusively in the human population. And so this predisposition to disease, we can estimate happened about 250,000 years ago, and it happened precisely in the route that leads to modern day humans after divergence from the human species from Denisova and Neanderthal. And so this particular change, this increase in copy, carries a gene known as BOLA2. It is the single biggest difference that it separates us from that of Neanderthals. And it carries within it a gene family known as BOLA2, which is important in terms of iron homeostasis within cells. We don't know all the details yet, but it's clear that patients that actually have these deleted are predisposed to essentially anemia. And it's possible that, in fact, the expansion of this duplication has been important in helping humans to adapt to maybe iron-rich or iron-poor environments compared to their antecedents. The irony is that this same event, this expansion of the duplicated sequence, is also contributing to the second most common cause of autism in the human species. So in summary, I've shown you that long-read sequencing is really revolutionizing our ability to access and characterize variation. So 73 to 75% of structural variation, particularly within repetitive regions, is being missed and not characterized by short-read Illumina whole genome sequencing. Complete telomere to telomere assemblies of human chromosomes are now possible, at least for complete hydatidiform moles. And in most cases, this involves using both long-read sequencing technologies, both Oxford Nanopore and PacBio. Combining HiFi PacBio sequencing technology with a linking read technologies such as Strandseq or a trio binning-based approach like parent-child trios allows us to create phased genomes that are 6 gigabases in size. This is an important achievement because it means that we're going to be able to systematically assess variation. So I'm going to argue that the future of clinical sequencing will not be whole genome sequencing with short reads, but will actually be phased and assembled genomes of patients compared to controls that identify the complexity of genetic variation underlying disease. And I believe the same principles will apply to our understanding of cancer genomes, although one could argue that considering the multiple haplotypes will be a much bigger challenge. So this is a small fraction of the people that have participated in the work that I presented today. I want to particularly acknowledge my postdocs, David Perubsky, Glennis Logston, and Peter Odbano, for allowing me to present unpublished work. I want to thank colleagues from the Telomere to Telomere Consortium, the Human Genome Structural Variation Consortium, and colleagues at WashU, as well as UCSC and USC, who are involved in essentially the Human Pangenome Project. So thank you for your attention. I'm happy to take questions if we have time.